Hi, my name is Joseph DeRisi, and I'm an associate professor at the University of California, San Francisco in the Department of Biochemistry and Biophysics, and I'm a Howard Hughes investigator. I'll be giving you a lecture today about our lab's efforts to develop drugs for the disease malaria. So first, a little background on the disease itself. It's a horrible disease caused by parasitic protozoa. It's characterized by intense fevers, chills, sweats, and in its worst form, cerebral malaria can cause death. Why do we care about the disease? Why is there a need to develop drugs? Very simple. The disease exacts an intolerable toll upon humanity, somewhere between 1 and 2 million deaths per year. That's someone who dies every 30 seconds. Furthermore, between 400 and 900 million people will come down with the disease per year. This causes a huge socioeconomic burden anywhere there's endemic malaria. So why is there a problem with this disease? Well, first of all, there's worldwide drug resistance. The limited drugs that we have, and I'll be reviewing those in a second, have become ineffectual in many parts of the world. Furthermore, we have no vaccine to date with an operational impact. And because people who live in endemic regions only acquire limited immunity, and that limited immunity requires repeat infections, we have only a limited capacity to ultimately control this disease. Okay, so first of all, why is there not major large drug efforts in malaria by the major pharmaceutical companies? There are some limited efforts, but for the most part, malaria occurs in the poorest regions of the world. Therefore, the economic incentive to develop drugs for malaria is not nearly what the economic incentive it is to, say, develop drugs for obesity or hair loss. Now, this disease is also transmitted by a vector, the Anopheles mosquito, and this complicates issues in controlling the disease. So, the disease itself, malaria, is caused by one of 4 parasites that infect humans, Plasmodium falciparum, the one that we study, and it's in fact the one that causes the most death and disease throughout the world. Plasmodium vivax, Plasmodium malariae, Plasmodium alveoli. These are all important parasites, but falciparum is considered to be the scourge of humanity. This is a blood smear of malaria. And basically, if you took blood from an infected individual and stained it with a stain called Giemsa that highlights where DNA is in cells, you can see that this cell here and this cell here are infected with parasites, whereas these healthy red blood cells here and here are uninfected. It is these parasites in the blood phase that we wish to develop therapeutics for to kill. So quickly, let's review the whole life cycle. Infection by malaria begins with the bite of a mosquito. Mosquitoes release from their salivary glands sporozoites, which travel through the bloodstream into the liver. It's here they infect hepatocytes. After going through multiple rounds of replication, these parasites will burst forth from the liver in a form called merozoites and begin to infect a cycle of red blood cells. The parasites invade the red blood cell, consume the hemoglobin within as an amino acid source, and then replicate in multiple rounds and burst forth again. And this process repeats itself again and again and again, essentially in perpetuity, unless the immune system brings the infection under control or the victim succumbs to the infection. Now, with some irregular frequency, parasites will develop into a sexual form called gametocytes, and it is this form that will be taken up by a new mosquito in a blood meal, and the entire cycle repeats again. So clearly there are several different places which malaria can be controlled. Malaria can be controlled primarily by wiping out the mosquito. That is, vector control can take care of most of the problem of malaria if you have the money, the geography to support it, and the social structures to support it. Vaccines seek to try and block the initial infection of the sporozoites into the liver. But as I said, so far there is no vaccine with an operational impact. Perhaps one day, but we don't have it right now. To date, all the success in controlling malaria, the disease, in people has been with chemotherapeutics, drugs. And most drugs target this asexual erythrocytic phase of development of the parasite. So let's quickly review malaria drugs as they exist today. Now, the most famous of all drugs is quinine. Quinine was isolated from the bark of a tree and was used by native South Americans and the Jesuits through the 1600s on. It's one of the most famous of all drugs because of its amazing potency against malaria. Now, one of the next most famous drugs is chloroquine. Chloroquine was developed originally by German pharmaceutical chemists, but then taken over by American pharmaceutical chemists during World War II. And after World War II became the drug of choice. Now, this was a wonderful drug. This drug was relatively non-toxic, was very cheap to make, and had extremely good potency. Yet the drug was misused. During the 1950s and 60s, there was irresponsible overuse of this drug. And as we know, when you overuse a single drug against a single target or pathogen, resistance can develop to that drug. And that's exactly what happened. So in many parts of the world today, chloroquine is very ineffectual. And so we need new drugs to bring a new generation of chemotherapeutics to bear on this problem. Now, after chloroquine came mefloquine. Mefloquine was also developed by Walter Reed, the US Army. And this was in response, in part, to chloroquine having resistance problems. Although mefloquine is a very expensive drug to make, and in a certain percentage of people causes fairly severe neurological side effects. So this Vietnam-era drug cannot be considered to be a cheap, widespread chemotherapeutic for malaria control. Now, after this was halofantrin. Halofantrin was again a US Army drug, and halofantrin was meant to overcome problems of mefloquine and chloroquine resistance. This drug, also developed by Walter Reed, suffers from certain cardiotoxicity side effects. Now, in the 1990s, we came to realize that artemisinin, a drug originally derived from Chinese herbal medicine, is probably one of our best hopes. Now, there are side effects with this drug as well, although currently there has not been very much in the way of resistance described for this drug. So, how do we go about developing new drugs? And what are the considerations we must take into account when developing them? Well, first of all, the cost of goods must be cheap. If we're going to make a drug for malaria, we have to shoot for a target of about 10 cents a curative dose. Any higher than this, and the populations that need it will not be able to afford it. Furthermore, the time to resistance of new compounds must be slow. The money and effort we put into drug development, only to have the compound become useless in the field very rapidly, is obviously not a very good effect. The other thing is, side effects must be minimal. So toxicity profiles are extremely important. And first of all, and most importantly, we must realize that the era of monotherapy is completely over. We know from HIV that treating with a single drug only breeds resistance quickly. And if we use drugs in combination together that target different areas of the pathogen, resistance is much slower to arise. And so, we don't just need one new drug, we need several new drugs, and we need to develop them in combinations. So, my lab is taking two different approaches to anti-malarial drug development. There are many labs and companies around the world that are developing malaria drugs, although not as many as I'd like to see personally. And so I'm only going to be describing our small efforts to develop anti-malarial chemotherapeutics. There are two approaches I'd like to tell you about. So the first is a parasynthetic chemistry approach, the so-called privileged scaffold approach. And the second is another approach called diversity screening. That is, why don't we look for new antimalarials amongst known drugs? But first, before I tell you about these two efforts, I have to tell you about the assay that we use. Any drug screening effort or drug testing effort always begins with an assay. And the assay here is a growth assay. Now, as I told you, the intraerythrocytic cell cycle of malaria is what we target with most drugs. During the blood phase of the malaria infection, this is where all the clinical manifestations of the disease actually occur. So this is the phase we want to target. It's a 48-hour cycle. It begins with infection, as I mentioned before, by merozoites. These merozoites develop into the ring stage inside the erythrocyte. There's a main metabolic phase in which the parasite grows. Multiple rounds of replication occur, and then these little parasites burst out and this cycle begins again. And so what we do is we treat parasites beginning here in this phase. We let them go all the way through this cycle, past merozoites, and into the next cycle, 72 hours. And what we measure is the increase in the number of infected cells over this 72 hours. A good drug, if it kills parasites, won't go around this cycle, and therefore we will not see an increase in the number of infected cells. So that's our goal. How do we measure the number of infected cells? Well, you could stain cells and count them, much the way these cells are stained here, but that would be laborious, time-consuming, and if you didn't count a lot of cells, could be statistically inaccurate. So we use a flow cytometer. A flow cytometer is nothing more than basically a fancy fluorescent microscope in which cells pass the objective of the microscope one at a time very fast. In this case, about 30,000 cells per second. And this microscope has the ability to count and measure the fluorescence in each cell as it passes the objective. What we do is stain cells with a fluorescent dye. Erythrocytes, blood cells, have no nuclei and no DNA when they're mature. Parasitic or parasitized red blood cells have DNA. So if we stain with a fluorescent dye, those cells will be fluorescent, and the flow cytometer will be able to count those and differentiate those from non-infected cells. So if we look at the data from the flow cytometer, here's a graph of uninfected cells, and this is a histogram. This is the number of cells on this axis, and this is the fluorescence. And so here's all the red blood cells with minimal fluorescence. Here's an infected blood cell population. So you can see here, here's the non-infected cells, and then here is the infected population. And you can clearly separate these two peaks. These are the cells here that we actually want to count. And we can just count those cells up as a proportion of the total, and we'll get an absolute number on the parasitemia. That is, the percentage of cells that are infected. And what we want to then do is just measure how many cells are before we treated with the drug and after we treated the drug relative to a control with no drug. Now, we don't put in tubes of these cells one at a time because we wanted to actually do thousands of tubes. So we developed a small program to drive a robot that actually feeds the cells to the machine in an automatic fashion. So the little robot is shown here, and all the cultures that we do are in 96 well plates, and this little sipper will come and get cells from each of these wells and inject it into the machine here. So let's watch a little movie of this in action. So what you can see here is the machine beginning to take up cells from each plate and inject it here. Then it goes through a loop and into the flow cytometer. And while those cells are being counted, this machine goes and gets another well and injects it in, and this repeats over and over again. So it takes about 12 to 13 minutes to complete an entire 96 well plate. And we can have several 96 well plates on deck to process. So it makes the process of counting multiple replicates of drug treatments very fast, very accurate. All right, so now that I've told you about the assay that we use, I want to tell you about our approach. So the first approach I'm going to tell you is the so-called privileged scaffold approach. And this entire project is a close collaboration between my lab and Kip Guy at St. Jude and a very talented graduate student, Peter Madrid. So the privileged scaffold is very simple. I told you that chloroquine was one of the most famous drugs, extremely potent in its day, very not toxic, extremely cheap to make, excellent drug overall. So who's to say that the scaffold that chloroquine is built on, the so-called 4-aminoquinolines, is a scaffold not worth pursuing further? That is, there may be derivatives of chloroquine, modifications of the basic scaffold design that could produce drugs that are just as effective. And so, if we just take a generalized structure of the quinoline series of compounds and we begin to systematically modify the quinoline structures in new ways that haven't been reported before, perhaps we can find new activities that will rival those of chloroquine. That's the hope, of course. And so, what Peter Madrid did is he started out with the basic scaffold and decided to derivatize, that is, put on different side chains and whatnot onto different parts of the molecule. So in this first example, what Peter Madrid did is he changed these two groups here at this distal amine. And a typical library that he made is shown here. So in this example of one parallel-synthetic library, Peter has kept one of the groups on this distal amine constant, a propyl group, and he has then substituted the other position on this distal amine with a variety of different side chains. And it is these different side chains that we want to test for efficacy against the parasite. And so a typical activity assay looks like this. Now what I'm showing you here is the different activities of all these different side chains as a function of how well they kill the parasite. And a dark blue means they're killing the parasite very well. So you can see there's two different rows here. The top row is a drug-sensitive parasite. The bottom row is a drug-resistant parasite that's known to be resistant to chloroquine. As you can see, here's our chloroquine control over here, and you see only a dark square at the top, not at the bottom. And you can see some compounds work just as well against both strains of malaria, whereas some other compounds, like these two over here, only work against the drug-sensitive version of malaria. And so obviously, we'll take the compounds that work against both, move them to the next phase, and then begin to try different side chains at different parts of the quinoline structure to, in a systematic way, iteratively optimize the compound. And so in this manner, we can create large numbers of compounds, each individually synthesized and optimized over several different libraries for the best efficacy against drug-resistant and drug-sensitive parasites. So one of the key aspects to this project is we don't want to waste money continuing on drugs that are never going to work. So it's the philosophy of fail early and fail cheap. We have help from SRI on this with John Marsalis. And basically, we'll test compounds for things like solubility, some of the most easy tests, genotoxicity using the so-called AIMS-2 test, cytochrome P450 inhibition to test to see whether these drugs will interfere with the metabolism of other drugs in humans. Permeability, this is a measure of how well the drug might be taken up by cells. This is a proxy for measuring basically bioavailability. Will this drug be useful as an oral drug, for example? Then we also want to know how well the drug partitions into red blood cells versus serum. This is important because in malaria, the parasites are in the red blood cells. We want the drug to preferentially be taken up in the erythrocyte relative to the serum. This is one of the beneficial characteristics of chloroquine. Now, one of the most important things to look at is stability. Taking human liver microsomes, they can be subjected to the compound, and we can calculate the half-life of the compound when subjected to these liver microsomes. These are basically, collections of liver enzymes that will metabolize a broad range of compounds. A compound that only hangs around for a couple minutes is not going to be very useful. Chloroquine, on the other hand, the model for these kind of drugs, has a very long half-life. This is important because when you give a dose of drug to a patient, you want enough compound to stay around in the body to have an effect on killing that parasite without having to redose that patient. Finally, a more expensive assay is the rat oral pharmacokinetics. How well does it get into the blood? Is it orally available? Does it have any toxicity against other organs, kidneys, and so on? And so, by these sort of preclinical measures, we can take these compounds and quickly weed out the ones that are never going to move forward because they have bad characteristics, and the ones that pass all these tests that can then move forward to the next iterative round of parallel-synthetic chemistry. Now, where are we today? So Peter Madrid came up with these top 4 compounds that have gone through several rounds of preclinical testing and iterative drug design. And these compounds all have very similar styles of scaffolds, as you can see, and all are extremely effective against both drug-resistant and drug-sensitive parasites, and they all have very decent half-lives. So obviously the next step of these compounds is to get them into more advanced preclinical trials, like studying cardiotoxicity and studying malaria infection in a primate model. If the compounds can pass these hurdles, if any one of the compounds passes these hurdles, they'll be moved forward, ultimately into phase 1 human clinical trials. That's our hope. Most drugs never make it there. Many compounds come in, few come out. This is the rule. But if we don't try these different compounds, if we don't pursue the different variations on the quinoline scaffold, we'll obviously never have a success. Okay, so basically that's one approach. The other approach is to shortcut the drug development process. How can we take collections of known bioactive or approved drugs and mine them for potential anti-parasitic activities? That is, maybe there are drugs in use today that are already good against malaria, that have already been validated for toxicity profiles, that are already known to be cheap and easy to make. Maybe they're generic drugs. So the goal is very simple. Acquire libraries of known bioactive or approved drugs and screen them for malaria. So this is the project of Jenny Weissman in my lab in collaboration with Kip Guy. And essentially what she took is a collection of about 2,000 different compounds that are bioactive or known to be approved drugs and screened them for malaria activity. And this resulted in a number of different classes. First of all, we found a number of compounds in the collection that are already known anti-malarial drugs. We're not interested in these because people have already studied them. We already know their anti-malarial potential. Now, there are a bunch of novel compounds that were not previously known to have anti-malarial activity. These are especially interesting to us. We can break these into three categories. First of all, the oral drugs. This is important because if we want to develop a pill-like drug, it has to be orally bioavailable. The IV drugs, intravenous administered drugs, and topical drugs. Now, topical drugs are not very interesting to us. Those are not going to be able to get into the bloodstream and so on, so we did not pursue these further. Intravenous drugs could be interesting, but they're not our highest priority. Oral drugs are obviously the way to go here if we want to develop a cheap therapeutic that could be taken in pill-like form. These broke down into several different categories, which I'll just highlight for you here on the screen. All of these, the ones that we chose from the oral category, had high therapeutic index. That is, the concentration that they killed malaria at was similar to the concentration that they're used at for their normal clinical purposes. And what we have here is a bunch of different compounds that really have very different normal uses. So propafenone is an antiarrhythmic drug for heart conditions. Perhexylene is a vasodilator. And these here are actually antipsychotic drugs. Now, propafenone is probably the most interesting drug to us because it's thought to block either calcium, potassium, or sodium channels. It's not clear which, and so we're very interested to see if propafenone is blocking a parasitic channel. And so more work will be done validating the target of this drug and developing new versions of the propafenone scaffold to test it against our model. That is, perhaps we can increase the efficacy of this drug and decrease its potential effects on the human channels. That's the hope, and of course that's going to take several years of parallel synthetic chemistry and structure-activity relationship. Okay, so our future directions here are quite clear. Basically, we want to engage in additional rounds of structure-activity relationship in both the quinoline series and the propafenone scaffold series. We want to screen additional bioactive compounds. There are hundreds of thousands of compounds out there that are known to have biological effects. We would like to mine them all for potential anti-malarial activities. We would like to validate and discover new targets. In many of these cases, like propafenone, we don't know what the parasitic target is. Validating the target would help us understand the drug, how it works, and potentially how to make the drug work better. Now, we also would like to investigate possible avenues of resistance. We know drug resistance can develop for chloroquine. It might develop for our new quinolines. How do we know what the resistance mutations will be if we, in a theoretical or hypothetical situation, overuse the drug? This is of primary importance. Also, we'd like to expand testing of multi-drug resistant parasites. The parasites we use in the lab today may not be reflective of what's in the field today. So bringing back parasites from Sub-Saharan Africa or Southeast Asia or South America to test in the lab against our compounds is of critical importance. And furthermore, for our advanced compounds, we'd like to complete preclinical testing and try and drive these compounds forward through the drug development process, ultimately in our best hopes to phase 1 human clinical trials. Of course, this is a long, arduous road and it's very expensive. But because academic institutions do not have the same profit incentives or profit motives that large pharmaceutical companies do, we have the freedom to explore these different avenues, to take early leads and late leads and explore them without the need to answer to stockholders, for example. So if you're in academic science and you're interested in malaria drug development, I hope I've showed you today that this process is actually very straightforward and that anyone with a moderately sized budget and lab can get involved in the drug development process without the need of major pharmaceutical backing. And so with that, I conclude this section on malaria drug development. Thank you very much.